Hello, everyone. Thank you for joining us today for the Human Cell Atlas Biological Networks seminar. The topics today are data submission guidelines and ethical considerations. I would like to remind you to join the HCA community and indicate your interest in joining one of the 18 biological networks. You can join more than one. Please visit our website and read up, and we're happy to have you. Please join. If you're preparing a publication that you would like to be included as an HCA publication, on our website, there's a description of the steps you can take to have this publication included. Please let us know. The upcoming HCA events. We have an HCA development and pediatrics meeting coming up starting on November the 20th. It's going to take place in Paris, but it's hybrid, so you can watch it on demand as well and register for online participation. And on December 6th, we're hoping to see you at the HCA White Town Hall that will be led by Sarah Teichman and Aviv Regev, and we will talk about the plans for HCA for the upcoming year, and we will welcome your questions there as well. So today's seminar agenda, we have two talks. The first one is by Kheng. He is at EMBL EBI, and he will talk about one place for all HCA data, the HCA data platform, and how we can support the Bionetworks. And the next speaker is Alexander Bernier. He is from McGill Center of Genomics and Policy, and he will be talking about practicing ethics and data stewardship in open biorepositories. And after their talks, if you are interested in asking your questions about ethics and data stewardship, please stay on this link and submit your questions via the Q&A function on the bottom of your screen. And if you would like to hear more about data submission or have specific questions, we will provide a Zoom link for you to follow. It will be concurrent session, office hours for data submission. So please, as you listen to the talks, decide which one you want to attend, and we'll direct you accordingly. So with this, I will gladly introduce our today's speakers. So the first speaker, as I mentioned, is Kheng. Wei is a bioinformatician and data wrangler for the Human Cell Atlas with a background in biochemistry, patient healthcare data, and genomic data. His work focuses on data consent and verification of data, which he is going to talk about in a second, with a focus on how to provide value to scientists and the scientific community. He's been working at the EMBL EBI as a part of the Human Cell Atlas data coordination platform for 2 years and drives their brokering of project expression matrices to analysis portals. Welcome, Kay.
Hi, thank you so much, Ellen. Can you see my screen that I'm sharing all right?
Yes, Wei, go ahead. Thank you.
Perfect, great. Thanks everyone for coming to this talk that I'm going to be giving about one place for all HCA data, the HCA data coordination platform, and how we can support the Bionetworks. Like Ellen said, I'm Kheng. I'm working here at EMBL EBI for the Human Cell Atlas, and I've been doing so for a couple years now. I'm going to be talking about this in 3 sections: an introduction to what the HCA data coordination platform is. Then I'm going to be talking about suitability and access is eligible HCA data coordination platform. And then finally, the data submission process. How do I start to submit to our platform?
Your sound is breaking up a little bit. Do you have a microphone or headphones you can use?
Yes, give me 1 moment.
Thank you. Sorry for interrupting.
How's that? Is that any better?
Much better. Thank you. Go ahead.
Okay, great. Yeah. Great. So the HCA data coordination platform really came out of a need for the Human Cell Atlas itself. As I'm sure you all know, the Human Cell Atlas involves creating comprehensive reference maps, and it's a global effort requiring hundreds of labs, organ-specific data, and lots of different disparate experimental techniques and data types. Every lab does things slightly differently, stores information and metadata and data in a slightly different way. And apart from that, there's also the different biological networks and community teams building tissue atlases. And it really came from this question: how do we bring all the data together so that anyone can find and use it? And from that, it really came to the specific principles of fair data. How do we make this data findable, accessible, interoperable, and reusable? We're moving much further away from an era of data generation, much more to an era of data management. Generating data, especially gigabytes and terabytes of data, is quite straightforward now, but it's much more about how do you make sure that data is still findable? How do you make sure it's still valuable to others rather than being locked away in silos in databases or in servers which aren't accessible or aren't findable and processable by other scientists and the community as a whole. So the HCA data coordination platform intends to store and provide single-cell data contributed by labs around the world. Anyone can contribute data, find data, or access our community tools and applications, and we try and make this data fair and comprehensive and openly available, and especially the metadata standardized. So I'm going to be showing a couple of quick sort of views and screenshots of our data portal itself. The link is here, and I'm sure you'll all be able to explore it in your own time. Apart from that, I want to briefly talk about the collaboration that we have in the data coordination platform. We work together with our friends and colleagues at the Broad Institute, which governs the data portal, storage, and retrieval aspect of the DCP, and also at the University of California, Santa Cruz, which work on ingestion and curation together with us at EMBL EBI. So this is the view of the data coordination platform at the moment. We've got 33.3 million cells, over 293 different projects, 485 labs. We've got community-generated multi-omic open data. And you can come to this platform and filter through whatever data and whatever selections you might want. Perhaps you are looking for single cell data for the blood, and you're looking for single cell data for the blood specifically from female patients who are of a certain age. You can search through and filter through our 33.3 million cells for the specific data that you're looking for that can help answer your research question or help give you more insight into investigating whatever it is that you might be interested in. We have lots of different projects, and it's all freely available there for you to explore at your own leisure. Now, you might be asking, why submit to the Human Cell Atlas data coordination platform? And I've got two answers for you. The first one is for an individual researcher or for an individual lab itself and for the community. In terms of why we can help you to archive your data and give you an accession, which many journals require. We can also help you in supporting and pulling together and structuring your metadata. We have lots of experience in creating a schema for metadata about how to structure all of this metadata that you might have about the donor, about the sample, about the protocols which went into your experiment, about how all of this might be structured, and also we can consistently put that in the same structure every single time. We also have an understanding of what metadata might be important for other scientists and what metadata might be important for making sure that data is reusable. For the community, we really are a specialized portal for 10x and single-cell RNA sequencing, single-cell data in general. We aim to be comprehensive. We aim to be high quality in terms of our metadata, and we are standardized in how we store all of this, which really allows for an integration of many different projects. So you can integrate multiple projects together and bring all of that disparate information together. We really believe that this integration between these different cell atlases, between these different groups, will really allow insights that wouldn't have been previously available. So we aim to be a comprehensive, high-quality store of single-cell RNA sequencing data, which allows you to save a lot of time, but also allows that really in-depth integration for those insights. I'm moving on into the second part of my talk here, which is on suitability and access. So in terms of the suitability criteria for submitting to the Human Cell Atlas DCP, we have 5 primary criteria. I'm going to be talking about the first 4 quite briefly, and then the last one in a bit more detail. So our 5 criteria are the organism, the sample type, the technology, the health status, and the access type. The first one, organism, is very straightforward. We primarily take data from human and mouse organisms. We've left the door slightly open here with other asterisks in case you have a particularly interesting or valuable single-cell experiment with other organisms. Feel free to reach out to us. I'll talk a bit more about how you can contact us at the end, but we almost exclusively take human and mouse organism data. In terms of sample types, we take data and metadata from primary tissue, organoids, and cell lines. In terms of technology, we focus on single-cell RNA sequencing, as I've mentioned before, but we also accept spatial transcriptomics such as Visium and NanoString, and supporting assay types such as ATAC-seq, ChIP-seq, bulk RNA sequencing, and others. In terms of health status, we previously focused a lot on building a reference map of healthy tissue, but we also see the value in having single-cell RNA sequencing data from diseased tissue, and so we accept samples from both healthy and diseased tissue right now. I'm going to move on and talk about the 5th criteria that we have, which is access type. Data can be broadly divided into 2 categories here, open access and controlled access. The thing to remember here is that in the eyes of the Human Cell Atlas DCP, data from live donors, or which has been consented for controlled access, is controlled access. Data which does not come from living donors or comes from non-human organisms is open access. What this means is that we can take data and metadata, which is open access and controlled access, but what we can take and the level of in detail we can take from differs slightly. So if your data is open access, then we can host your raw sequencing files. We can take your analysis files. We can take metadata on the donor and the sample level. So that's the donor's age, sex, gender, smoking history, ethnicity, medical history. We can go quite in depth on that level. With controlled access, with data from living donors, we can only take analysis files and metadata at the sample level. So we can't take any identifiable data or donor data. We can take, however, metadata on the organ part, what protocols were used to collect, to process, to dissociate or enrich these specific samples. There is a slight caveat here with living donors in that if metadata is already publicly available about the living donors, then we would also look in those repositories to take that metadata and also host it on the Human Cell Atlas data coordination platform. I'm going to talk about my 3rd and final part of the presentation, which is on data submission to the HCA DCP. So the data coordination platform data flow can really be broken down into 3 major parts. Labs contribute single-cell data, the DCP curates and uploads authors' data, and researchers access that data on the portal. And I'll go through these step by step. With the first step, labs contributing single-cell data. We have a suitability checklist, and that suitability checklist is essentially what I had just gone through, that table which I had gone through previously, ensuring that your data and metadata comes from human or mouse, ensuring it's the right technique, ensuring that it comes from normal or diseased tissue, ensuring that it's either open access or controlled access, and understanding what that means. Once you've gone through our suitability checklist, then we have
two separate forms here. We have a terms and conditions form, which talks a bit about the access requirements and the accessibility of your data, perhaps what state your data is in. And we have a questionnaire, which tells us more about the scientific background of your experiment. Were there any cell lines? Were there any organoids? What organ was used? What technology was used? And based off of those two forms, we get a fairly good understanding of how we might help you to contribute your data, because every experiment is different, especially at the single cell level, and every experiment will be modeled slightly differently. So if you would like to start contributing data, a great place to start is by filling in the terms and conditions form, the questionnaire, and or sending us an email at wranglerteam@data.humancellatlas.org. Once that has been done, we go on to the second step, which is the DCP curates and uploads authors' data. This is normally the largest, most time-consuming step because it involves a lot of collaboration between us and you, the community, the scientists, the researchers. It involves some elements of back and forth and understanding on a really fundamental level what the data and metadata you were working with means. It involves curating the metadata, obtaining important bits of metadata that would mean that the data and metadata are fair, that it is more reusable. It involves also creating your analysis data. We offer a service where we can wrangle your analysis data into other analysis portals such as Cell by Gene and other locations. We also have our own in-house validation, which validates your metadata using controlled ontologies and controlled vocabularies. This also validates based on the raw data and the FASTQs we accept and the analysis data. Once we've curated all of the metadata, the analysis data, and validated it all to make sure it fits our standard and a high quality of data, then we can upload this data onto the Human Cell Atlas. That essentially completes the submission. If you wanted your data to be archived, we can do that and provide accessions to you specifically, which you might require for your journal. We also acknowledge publicly the contributors and the people and the scientists who have made this data set possible, and that's all openly available and openly acknowledged on the data coordination platform. And then your data and metadata is publicly accessible to the rest of the community. And then your data is now available. Researchers can access the data on the portal. This includes being able to filter for data using HCA metadata. I showed the screenshot slightly earlier about being able to search on donor info or tissue types or specimen info. You can search on free text searches. You can search by the file type you're interested in the analysis files, and many, many more other methods of filtering. And that's all available on our portal, which is the data.humancellatlas.org. Apart from the data coordination platform, we also work alongside other analysis and data portals. I have a link here to the Human Cell Atlas data portals, humancellatlas.org/portals. We, as I previously mentioned, wrangle analysis files to Cell by Gene, and UCSC Cell Browser will add your data there for you in the appropriate format according to their specific requirements. We also work closely and in collaboration with the Single Cell Portal, the Single Cell Expression Atlas, and Cambridge Cell Atlas, and we really work together to try and fulfill whatever requirements and needs that the scientific community has. I'm going to finish off by talking a little bit about our focus as the Human Cell Atlas DCP. We've always been aimed to be an open and comprehensive portal of single cell data, and in the past this has meant wrangling data sets from direct contributors here at the Sanger and all around the world, but also means that we keep an eye on the literature, and if there's an interesting or valuable single cell paper from the literature, then we will take that and wrangle that into the Human Cell Atlas data coordination platform as well. We were recently at the HCA Vienna General Meeting, and we really saw the energy shift with the Biological Networks group attempting to set up version 1 atlases and focusing really heavily on that. And so we pivoted as the DCP to focus more on the biological networks. We are currently working with the lung, immune, kidney, skin, and development, quite a few of them. biological networks, and we really aim to develop a common standard for integration through working closely with them. We curate their metadata, we're identifying common data elements between different biological networks. We're working through issues around sharing and a lot of these thorny issues which are existing around there. Our aim is to make the biological network data available, but also standardized and available for integration, which a lot of the scientists and a lot of the other groups are specifically hoping to do. Our DCP data grows with each monthly release. We have 33.3 million cells at the moment, and we would love for you to be part of that effort. Let's build together. Questions and feedback, and your community is really what we need here. We need to engage with you and figure out how we can best help you do what you do faster and better and in a more efficient and high quality way. Funding and acknowledgements, our friends and our colleagues at the Broad, at the UCSC and at EBI, we're funded primarily by the CZI, and this is our fantastic team that we work with all across the globe. And obviously we're majorly funded by the CZI, but we're also funded in part by lots of different other wonderful and excellent programs which make us able to do the science that we do. Thank you very much.
Thanks so much, Wade. This was great. We do welcome questions from the audience. Please type them in the Q&A icon on the bottom of the screen. Oh, there's one comment from Gamal Akabani and also one question. The comment is, Gamal loves the graphics, so thank you for that. The question is, could you please elaborate about the cell type software and its limitation on immune cells?
So I can elaborate broadly on our efforts working together with cell typing, with the analysis portals that we work with, but I'm not specifically familiar with the cell type software. We try to do a host of cell type annotations together, working with the ontology team here at EMBO EBI and together with our friends at Cell by Gene, but I'm afraid I might not be able to specifically elaborate on cell type software.
Thank you. The next question is from Chris Tuggle. Can you comment on the support for non-human, non-mouse data?
Sure. So in regards to the support for non-human, non-mouse data, I spoke earlier about how we primarily accept data from human or mouse. We leave the door open for particularly valuable or interesting data from a non-human, non-mouse set. This really means that it works on a case-by-case basis. Reach out to us, send us an email, tell us about your data set. We'll start a conversation and we can see how that goes. Like, we have a great team here of wranglers who are all interested in different aspects of the science, and we would set up a meeting and work with you directly on that.
There's a comment for Giorgio Cotteretti. I imagine that the controlled data from the living human beings are because of the virtual impossibility to provide privacy for sequencing data. However, there are ways to limit this.
Yeah, I guess I can comment on that briefly. That's a really good point about the difficulty to provide privacy for sequencing data. We do work together with repositories which primarily work with controlled access data, such as the EGA. So if your data, your control data is stored in the EGA repository, we can add a link to that repository. We can add the specific accession, and that builds a link between all of the metadata, which is controlled but still available on the Human Cell Atlas, and the EGA, where they can reach out to that specific data access committee.
Thank you. A question from Mitnala Sasikala. Do you have data on pancreatic islets, healthy and diabetic?
Off the top of my head, I am fairly certain we have at least some pancreatic data. I can have a look at that, especially during the office hours that are coming up slightly later, and walk you through that through our data portal, if you'd like, Mitnala.
Thanks. And the last question is, could you please elaborate a bit more on what your policy is regarding data from live donors and including relevant metadata such as age, sex, etc.? You mentioned that it is already publicly available. If it is already publicly available, you will accept this metadata. Could you please clarify? Thanks.
Yeah, so you've pretty much hit the nail on the head there. In terms of what I've been sort of informed about the Human Cell Atlas and like our policy regarding data, it's that if it comes from living donors, especially living human donors, it falls under the category of controlled data. However, if information about that, if data and metadata is already publicly available, like in a publicly available repository, we would look in those places to bolster that the metadata that we have in the Human Cell Atlas. I can also talk a little bit more about this in more detail in the office hours, if that is still not super clear.
Maybe Alex actually.
And yeah, Alex will probably talk more about this, given that he's talking about the ethics
Sure. So just a really quick comment on that last point is that so you've hit the nail on the head there. Essentially, if the information's suitable to be released in full open access, then the custodian or the data contributor that deposits the data in the HCA can indicate that. And specifically, I'll get into these details a little bit more during my talk, but essentially, if the data is subject either to an informed consent or to another authorization, another legal justification for depositing it in full open access, then that can be indicated and the data can go in the open access portion of the repository. To help make that determination, it's ultimately left to the institutional legal counsel and the other personnel at the depositing institution, the researchers. But to make that determination, there's a set of core consent elements that are available on the HCA ethics website. If you go on the HCA website, there's a little tab that says ethics, and you can find core consent elements there. If those elements are met, it's generally either through a consent form, another mechanism, it's generally appropriate to deposit in open access. If those are not met, but other permissions that allow for deposit in controlled access are met, then the data can go in the controlled access prong of the DCP. And there's still internal discussions that are occurring that haven't quite been finalized regarding exactly which data elements are in controlled access, which are in open access. But that's the general position as it stands, but at least by my current understanding.
Alex, thanks a lot. And Wei, thank you. I guess this is a good segue for me to introduce Alex formally. Alex Bernier's his research is primarily concerned with data protection law, as you probably understood. He is at McGill University, and he's a part of the Center of Genomics and Policy. So data protection law, open science, and research infrastructure is what Alex is studying. He is a member of the European Canadian Cancer Network's International Ethics Board and the ethics officer of the Canadian Open Neuroscience Platform Ethics and Data Governance Committee. So Alex, go ahead and share your screen and let's get started.
Brilliant. Thank you so much for having me. All right, so let me get my screen shared and dive right into it. Okay, can everybody see these slides?
Yes, perfect. Presenters, yeah.
Okay, wonderful. So today I'm going to be discussing with you some more general issues around open science and specifically the deposit of data in open biorepositories. So my colleague Wei got a little bit into some of both the technical issues, but also the public policy issues inherent in depositing genetic information and other forms of health information in totally open access repositories. I'm going to start with some general examples and some examples from other consortia. And then I'm going to segue into some specific guidance that's being issued in the context of the Human Cell Atlas. So the first question we have to answer is essentially, what are the stakes in determining how information can be used and in determining the kinds of policies, the kinds of governance rules that are applicable to open access repositories, whether they be fully open access in the sense of totally publicly available, as are some portions of the Human Cell Atlas, or whether they be controlled access resources that an individual would need to apply for access to. And what some of the major stakes come from this issue known as legal or normative interoperability. So just like how in a technical context, we think about interoperability, and I'm not a technical person, so I may get the jargon wrong, but essentially we think about things such as compatible file formats, compatible methodologies for describing certain features and data. We also think of compatible, sorry, can everyone still see my slides? My computer seems to be acting up a little bit. Can you still, just a moment?
Actually, it paused for a second.
Yeah, okay, let me share that again. Sorry, there was some kind of technical issue on my side of things. Can you see them? Can you see this again now?
Yes, okay.
Okay, brilliant. Yeah, sorry about that. All right, so just as you can think of technical interoperability in terms of formatting files, formatting data in ways that they can be used for compatible purposes or that different systems can communicate with one another, these same similar issues come up in the context of normative rules. One of the first reasons behind that is that different rules that are applicable to how information data can be used come from different sources. And the second comes from the fact that there are different levels of sensitivity or different ethical issues inherent in using kinds of data that come up from different contexts. So to make that a little bit more tangible, when we think about a large-scale consortium such as the Human Cell Atlas, it's structured across a large number of different jurisdictions, and each country might use different mechanisms such as clinical trial regulations, data protection laws, open information, or access to information legislation to determine how locally generated information can be used on the one hand. And then on the other hand, the sensitivity of specific populations might mean that their information needs to be safeguarded in different ways. So for instance, pediatric populations might be subject to a different profile of risks than adult populations on the one hand, or genetic information might create different ethical challenges than does population health data. One of the questions that we need to think about is how can consortia respond to these concerns, and how can they do that in a way that makes sure that information can still be used and leveraged at scale. So for example, one might think that a simple solution to these challenges of governing data according to local norms and according to the specificities of the kind of data being used would be to ensure that each institution develops their own appropriate governance policies. And in the context of individual studies, this is something that happens all of the time. Local institutions will dialogue with their stakeholders, the affected communities and populations on the one hand, jurists, and even research ethics committees, other such stakeholders, and will formulate or develop a governance program, a way of stewarding that data that responds to local needs. But one of the tensions or challenges that we often encounter in developing this for large-scale consortia is that there's really two needs that are being served. The first is to ensure that the information can be used in a way that respects the applicable local norms, but the second is to ensure that the governance rules that are developed for such information are compatible, are scalable across all of these institutions, that they're meaningfully comparable on the one hand, and that they're similar enough to ensure that the data can be used for common purposes across all instances of downstream use. So for instance, we can imagine that if 100 data sets are generated at 100 different institutions, that if the contractual rules, the terms and conditions that applied to the use of each were vastly different from one another, that it would create first an issue of incompatibility, but also a need for human review in ensuring compatible use that would outstrip the available resources to all of these communities. That is to say that it wouldn't be possible for an interested user to read all of those policies and to determine, well, does the information I want to use correspond, do the governance rules applicable to the information I want to use correspond to my intended use, or not. And here we see a set of some of the kinds of rules that can be applicable to data and determine whether it can be used or whether it can't be used for a particular purpose. It's also interesting to note that some of these are left to the discretion or the decision-making of the institution that decides to utilize the data. So for instance, here we see things like private law contracts, intellectual property licenses, institutional policies that are within the control of the organization that determines how the data can be used, but others are not necessarily things that are liable to be changed, that are liable to be modified. So we can think of things like data privacy law, interpretive guidance, court decisions, things that are just out there and that states or regulators have decided upon and that can't meaningfully be changed by a group of researchers or a research consortium. One thing I'd like to add to that is that this concern with the non-compatibility of the governance rules applicable to multiple data sets is not just a theoretical concern. It's not just an issue that could arise. It's an issue that does systematically arise in large-scale consortia when researchers attempt to repurpose information that has already been generated for a new purpose, but don't prospectively, so from the outset, ensure that the governance rules applied to the data and reflected in documents like informed consent materials, contracts regarding data use, and governance policies are compatible with one another. So here we're drawing from an interesting prior example. This is the EU CanShare Consortium, and it was an effort, I guess around 2018 or so, to repurpose a large number of data sets from distinct population health consortia in the European Union and also a few in Canada. Now, one of the interesting things here is that because these are population health cohorts, the information was generated across a long span of time, many, many years, and the governance rules applicable, of course, were developed almost in a piecemeal way as time went on, because, of course, these different cohorts, the data wasn't generated with the intent of bringing them all together. These were individuated, localized examples of population health cohorts, and it's only in the contemporary age, in the mid to late 2010s, that it was decided to bring together the information for further downstream use. So there, of course, wasn't this prospective effort to make sure that the data use conditions applicable to each were compatible. And so the effort that was done was essentially an assessment of how compatible the data governance rules applicable to each such population health cohort were. And the green here generally means a high degree of compatibility, a high degree of data openness. Yellow means ambiguities or a lack of information, but again, no information that really suggests that use is limited or precluded. Orange here refers to situations in which a particular set of uses might be limited, but there's no serious restriction or serious preclusion. And of course, the red is demonstrative of some kind of preclusion limitation. So far things look good. The possibility to use this data for compatible downstream purposes appears to be fairly compatible. However, the interesting thing is that once we move to this information here, which details a set at the top of governance conditions, governance rules on a data set by data set basis, and again, a sort of grading or shading from most open to most restrictive, we start to see that the restrictions, the limitations in information use are highly divergent from cohort to cohort, but also very heterogeneous, that some cohorts allow certain things and others different ones, and there's not that much compatibility. Here's another set of diagrams which reprise the same information in a different format. Unfortunately, there's a color version of this. This is in black and white, so it's not terribly clear for the reader, but it does show, and you can tell by looking, if you follow one particular cohort relative to the others, the choices in shading are very, very different from one to the next, which essentially means that the governance rules are very different from one consortium to another. And we can imagine that there would be challenges for a principal investigator applying for access, downstream access to this information. We can imagine that they would face challenges in accessing all of this information for compatible uses, or simply in determining how broadly this information can be used. One interesting question is, so we see that this happens in this particular consortium, but we're looking at one population within Europe, within the population health area. It's a very, it's a very distinct example. Would these results be generalizable elsewhere? There's some other research that was done with a consortium called REACH, and this is a group in Canada principally that looks at, so it's again population health, but it's within the context of these pediatric mother-child birth studies. And what we found, and you can see because the percentages differ wildly from one particular set of uses to another, is that whilst studies generally allowed the reuse of information, the transfer of information, and we can see because at the top right over here, the percentages are in the 90s or the high 70s. So again, we can see that most institutions enabled the reuse of their data, but the more we scroll down this graph, the more we see that the conditions according to which the information can be reused differ from one organization to another. And there's some other data sets, which I won't bore with you with at this particular instance, but there are other data sets that really dive into these within the REACH effort, that is, that really dive deeper into these conditions and do show that the governance rules generated by independent organizations not working together, not coordinating their efforts, can lead to data that's very rich, very valuable, but isn't necessarily interoperable. From that general framing, let's turn specifically to the tools that have been developed in the context of the Human Cell Atlas. So on the Human Cell Atlas website, and I do believe that these tools will be highly useful to some of the people here in attendance, there are informed consent templates and self-assessment checklists, which are intended to ensure that so long as they are used to generate informed consent materials and followed in general terms, the output data will be subject to conditions of use that allow for its interoperable downstream use. And specifically that the conditions of use will be as broad and open as is possible. So we'll allow for things like cloud storage, international transfer and sharing of data, downstream use for a broad range of health research purposes. So the information can be used broadly, and also that the terms of these informed consent documents are, generally speaking, compatible. Such tools were developed by Emily Kirby, who's also on the call this morning, and a number of other colleagues at the Center of Genomics and Policy, myself included. And they are addressed to a number of different populations. There's a set of tools for, of course, adult populations, a number of specialized tools for distinct scenarios of deceased populations. And there are tools, a slew of tools directed to pediatric populations. There's also a set of additional tools which are not informed consent materials, but really are additional documents that can help researchers determine whether or not it's appropriate for them to deposit data in the Human Cell Atlas DCP, or what additional measures might need to be taken in order to deposit such information. So one example thereof is a set of core consent elements, which are, as we discussed earlier, these core permissions that we hope all Human Cell Atlas data will align with. And then what's referred to as a retrospective consent filter, which is essentially a self-assessment tool to determine whether legacy data sets, so data sets that were produced prior to the advent of the Human Cell Atlas, or were produced with a different research purpose in mind and would subsequently potentially be contributed to the Human Cell Atlas. There's also a set of training materials to ensure that recruiters who are recruiting and possibly also performing studies use the consent tools and navigate the informed consent process in a way that ensures that research participants have the opportunity to ask necessary questions and are well informed as to what their participation will entail. Here, and this document I think might prove particularly useful, we have a set of permissions that are necessary on the left side to deposit data in controlled access. So in the managed access prong of the Human Cell Atlas DCP. And on the right side here, we have a set of criteria for the deposit of data into open access. Generally speaking, this notion of open to controlled access recognizes that certain data sets will be more sensitive than others, and due to either legal rules, which determine how the information can be used within a particular jurisdiction, institutional policy choices made determining how openly information can be shared, or even simply the preferences of research participants, that all of these elements can determine when the data is acceptable to place in open access versus those circumstances in which it would be best to leave it in controlled access. Here we also see a mention of a registered access tier. The Human Cell Atlas is not at this time intending to utilize a registered access tier, but certain consortia will place data or tools in registered access. And this is usually done in those circumstances where the information is generally acceptable for public release, but there are certain residual risks regarding individual re-identification or group level stigmatization that militate for some measure of access control being put in place. So what we can imagine, for instance, is certain genomic beacon style systems or query engines that basically run requests, run analyses across group level data sets. But certain organizations might decide to put these in registered access rather than fully open access out of concerns related to strategic re-identification attacks that target particular genomic variants. And then the use of registration can enable the use of query budgets and other technological safeguards that preclude re-identification. My last slide here. So this, we see that it refers to the International Cancer Genome Consortium rather than the Human Cell Atlas. This is from previous particular consortium. But it shows how in certain instances managed access is practiced, managed or controlled access. So in a controlled access context, it's customary to have two institutions, a data access compliance office on the one hand and a data access committee that are responsible for adjudicating requests for downstream data use according to specific criteria. So this doesn't resemble ethics review. It's not an ex post interrogation of a research group's research aims. Really what it intends to do is quite simply make sure that researchers that are intending to access data for downstream use have the necessary institutional credentials, that is to say, they have a PI role or another role that enables them to use that information that shows that they are capable of conducting the kind of research that they intend, that they're affiliated with a bona fide research institution, and also makes a few punctual verifications of the intended purposes of data use. So things like, do the researchers have a legitimate scientific project? Are there glaring risks of individual re-identification or of group level discrimination that are really baked into the research concept? And once that's done, the role of these organizations is to bind those institutions to contractual agreements that give users access to that information according to terms that protect the privacy, confidentiality, and interests of the research participants, of the individuals concerned by the data. It's the intention of the Human Cell Atlas to implement a data access committee to govern, and a data access compliance office to govern access to the data that it hosts. The details of that are still being determined, and the timelines for that remain undecided. But that is the eventual hope, that this would be the approach adopted in the HCA. I think I'm running up against time here, so I'll leave it at that. I wanted to thank you all for your attendance, and I'd be very glad to take any questions or even to discuss informally outside of this context. We also have an EWG specific email address, which I'll circulate, that you can use to reach Emily Kirby, Bertha Knoppers, and myself with HCA related ethics queries.
Thanks so much, Alex. So please do submit your questions to Alex in the Q&A section at the bottom of your screen. And maybe actually while we're waiting, it's a good opportunity for me to introduce Emily. Emily, do you mind coming on screen as well? Great, thank you. You can unmute yourself this way. We can see you side by side.
Yes, many years of Zoom, I still don't remember doing it.
Yeah, great. Okay, so our ethics experts for the last part of this webinar are both from McGill University Center of Genomics and Policy. And Alex is joined today by Emily Kirby to answer your questions. She is the academic coordinator of the ethics working group of the Human Cell Atlas. She currently works on the development of ethical and legal documents and tools used to facilitate policy interoperability and data sharing in the context of data-intensive research, omics, clinical data, etc. Emily has been involved in examining ethical, legal, and policy issues in a number of Canadian and international data sharing initiatives. And let's see if there are any questions to Alex for his talk. Doesn't look like it. So then I will introduce Gabby as well. Gabby, would you like to come on screen?
Yeah.
Oops. I guess it's because you're sitting next to A, right? Yeah, there's an echo. But so as you figure, Gabi's sitting next to A. Both of them today will be answering your data submission related questions. We will provide data submission office hours link in the chat window. Gabby is a user experience specialist at the HCA data coordination platform team, and she's also based at the MBL's European Bioinformatics Institute. So what we're going to do is, you see the link, please copy it if you would like to attend office hours for data submissions. If you're staying here, please prepare your questions for Alex and Emily. Meanwhile, I will stop recording, and for those of you who tuned in to see us via YouTube or Bilibili, thank you very much. Please join us for other biological network seminars. I would like to thank, actually, let me show my thank you slide. Yes, so I would like to thank everyone for joining us today, and thanks to our speakers, and thanks for the Chan Zuckerberg Initiative who made this series possible. So with this, I'm stopping the recording.
Thank you, Ellen. Just a brief note, I see that Gamal has put a question into the chat. I wonder.
Oh, cool. Great. Okay, let's read this and answer this, and then I'll stop. Okay.
Yeah, happy to, whether it's Emily or I, for one of us to take it. Here the question is, Alex, the system is geared towards well-developed nations that can establish these procedures. How can we help other contributing nations that do not have such infrastructure for data entry? So I think there's a lot of really critical and interesting insights baked into this question. I know that Emily and another colleague, Michael Beauvais, at the outset of the initiative had essentially done this landscape mapping exercise that looked at the regulatory requirements that are applicable in a number of different nations really around the world. It was an incredibly extensive review of the legal requirements for things like tissue sampling, data contribution, and so forth. And so here we can think of two sort of sets of issues that come out of Gamal's question. The first one being, of course, I think the easier question to answer, which is, there are different regulatory requirements in different nations. But of course, the second and the more challenging one, and I think, what we're really trying to get at here is, what about capacity? So in the sense that institutions that are possibly, whether they're in the global south or they're outside of major metropolitan research centers don't necessarily have specialized staff who are accustomed to dealing with bioethics and the data management issues that come with that. Anything that's from a technical standpoint, I assume that, you know, similar issues will arise, but it lies outside of my personal disciplinary experience. So two things that I would say to that is that in terms of reducing some of these capacity burdens, the long-term the long-term answer to that question is that there will be investments in developing local capacity in technical terms, but also in terms of bioethics expertise and navigation of regulatory requirements, that investing in those capacity building efforts is a key long-term objective. In terms of the short-term things that can be done to facilitate this process, the tools and templates that are developed, they're not a one-size-fits-all answer, but they do reduce some of the burden on institutions to develop their own governance-related materials. And we've had certain specific contexts where researchers from jurisdictions that had very specific governance-related issues, so for instance, their local institution or their local law, placed limitations on how the information could be used that were outside of what was addressed in the available materials. And we did engage in dialogue with those researchers to see how we could better map the existing tools to their particular needs. So one answer there is collaboration between the ethics working group and between particular populations. But again, you know, these aren't perfect answers, and I don't think we have perfect answers yet. I think it's a very fair comment, and I think it's something that needs further thought.
If I can,
Okay.
yeah,
Yeah,
if
go
I
ahead.
can jump into that and add to it as well. So I think Alex exactly made a very good summary of saying it's not one size fits all. I wanted to point out also that there is within the HCA an equity working group that works also on capacity building and sort of does a lot of road shows with different regions and tries to sort of get feedback on definitely technical limitations and how to sort of overcome some of those challenges. And again, as Alex mentioned, the ethics toolkit that's on the HCA website does have a few resources to help with how to explain some of the notions underlying the HCA, such as, you know, consent to open sharing, etc. So again, they're not a one size fits all. They're not perfect for every nation, every country, every set of rules, but they do try to provide a very simplified view of, you know, what is the HCA, what are the ethical considerations. And the last point I did want to mention is that within the ethics working group, so it's a group of about 20, you know, has 20 members. We were very mindful of trying to have members from different regions that would have different approaches to research ethics. And we do have some regional network liaisons within our group. So for HCA Asia, HCA Latin America, etc. So I think even Mayumi is our rep for HCA Asia is on the line as well. So we try to make sure that our group, the ethics working group, is involved with different realities, different data sharing environments, which would reflect in the tools as well.
Great, thanks so much. So for those of you who would like to stay and ask more specific questions on ethics, please stay. And for those who want to attend office hours, follow the link in the chat window. And thanks everyone. I'm stopping the recording right now.